Hello class, my name's Chase,
And I'm Johnny.
and this is our acetylcholine signaling project. So Johnny, what is acetylcholine?
See, that's a great question, Chase. Well, actually, it's quite simple. Acetylcholine is a neurotransmitter that's made up of acetyl-CoA and choline. And together, acetylcholine regulates cardiac contractions, blood pressure, and glandular secretion, which is a fluid released by glands in the body. It also plays an important role in memory, learning, attention, and regulating neuroplasticity in our brains. This means that low concentrations of acetylcholine are correlated with Alzheimer's and dementia, while higher concentrations of acetylcholine can cause sweating, diarrhea, cramps, and increased salivation. Alright, so here's an overview of the whole process of acetylcholine being synthesized in the basal forebrain and then going to synaptic vesicles and then eventually reaching and binding to G protein coupled receptors where the signal is amplified, and then you get your response, which is muscle contraction or anything else. But basically, starting the basal forebrain, your acetyl-CoA and your choline are synthesized together to create ACH, which is just short for acetylcholine. So labeled right here, ACH, those just represent the signaling molecules of acetylcholine. But through synaptic vesicles, that's where you're actually transporting these acetylcholines, and then through exocytosis, which is a process of outputting from vesicles, you release these acetylcholines outside of cells where they will seek and bind to G protein coupled receptors. And because we know that GPCRs are located on the surface of cells, um, we know that acetylcholine is hydrophilic because they do not cross the hydrophobic nonpolar fatty acid phospholipid bilayer right here. One benefit of acetylcholine being a hydrophilic signaling molecule is that it helps elicit short-lived responses, and these are done so when acetylcholine binds to the G protein coupled receptor, which activates them, and then it helps them amplify signals by signaling to secondary messengers to help even amplify this, to help transduct the signal even more by amplifying it and activating other proteins. And one con of this is that short-lived responses mean that they can only travel short distances compared to hydrophobic receptors, and those are basically receptors that can help elicit responses that are more longer lived but can carry throughout the body in more longer distances.
So first, the acetylcholine neurotransmitter binds to the special neurotransmitter binding site, which is right here in the dark green. And this is actually special to the metabotropic receptors because not every receptor has this. So once the neurotransmitter binds to the receptor, GDP binds to the alpha beta subunit, and once this has the GDP attached to it, it actually attaches itself to the beta and the gamma, and this creates an inactive trimer. The binding of an extracellular signal activates the metabotropic receptor, and GDP is then replaced by GTP, just like that. Once the GTP binds, the alpha subunit becomes activated, and the alpha unit disassociates itself from the rest of the complex. So something like this. Okay. And once it becomes disconnected, the beta and gamma subunit can actually attach itself to the ionotropic receptor, which is over here. So it will look something like this. And while that's happening, the alpha subunit attaches to the effector, and this effector acts as a secondary messenger, and it amplifies the signal to elicit a specific response. Like we mentioned before, the response is muscle contraction, glandular secretion, and it can also play a part in memory recollection. It all depends on the type of receptor protein that the ligand is sent to. And once this beta and gamma subunit attach to the ionotropic receptor, it actually opens up the channel so that way more ions can pass through, just like this. So Johnny, how is the signaling process terminated?
That's a great question, Chase. Acetylcholine esterase, also known as ACHE, is a powerful hydrolytic enzyme that is responsible for terminating the process by splitting up acetylcholine into choline and acetyl-CoA. ACHE is special because while most neurotransmitters are terminated through reuptake, acetylcholine is terminated specifically by this enzyme. It is the only neurotransmitter that is cleaved. ACHE is extremely effective and is the #1 way of inactivating acetylcholine. In the diagram, we have represented ACHE as the terminator with a pair of scissors, and it's basically cutting up the acetylcholine into acetyl-CoA choline. And because it's split up, the receptor on the surface of the muscle cell has nothing to bind with, thus terminating the signaling process.
Thank you for watching.
Thank you.
See you later, man.